Think the most famous scene in science fiction is a lie. For 30 years, we believed the fantasy. A mosquito trapped in amber. Dinosaur blood extracted from its gut. A theme park full of resurrected reptiles. The premise is elegant. The science is impossible. DNA has a hard expiration date. Under perfect conditions, frozen, sealed, protected from oxygen, genetic material survives a maximum of 6.8 million years. That is the ceiling. The absolute limit. Dinosaurs went extinct 66 million years ago. The gap is not close. It is not debatable. It is mathematically insurmountable. Every strand of dinosaur DNA unraveled into noise tens of millions of years before humans existed. But here is what does not make sense. If DNA cannot survive, if soft tissue is the first thing to decay, if organic molecules dissolve into entropy within geological seconds, then why did scientists find iron traces inside a 100-million-year-old feathered dinosaur tail? Traces that look exactly like fresh blood. That question has an answer. And it will disturb everything you think you know about preservation. Because that amber pendant on your dresser is not a pretty stone. It is not fossilized sap. It is not a gentle time capsule. It is a prehistoric crime scene. And the creatures trapped inside were not preserved by accident. They were executed by a chemical weapon. Resin did not want to preserve anything. It wanted to kill. The tree deployed it as an emergency response, a toxic, high-viscosity flood designed to seal wounds and poison invaders. The immortality that followed was never the point. It was a side effect of molecular murder. To understand what that amber contains, to see the bodies frozen inside, you need to understand what resin actually is. Not what you were told. What it does to flesh. Resin is not tree sap. Sap is nutritional fluid. It moves sugars. It feeds growth. Resin is something else entirely. It is a defensive secretion loaded with terpenes, volatile organic compounds that provide the medicinal smell and toxic taste designed to deter anything that bites, crawls, or drills into bark. Think of it as industrial superglue crossed with chemical mace. A high-viscosity bioshield deployed as an emergency response system for wounded timber. When a tree is damaged by a beetle, a storm, a claw, the resin flows. Thick. Stickier than tar. Denser than honey. And anything caught in that flow does not escape. The trap has no moving parts. It is pure chemistry. As the volatile compounds evaporate into the air, something irreversible happens. The molecules begin to link. Chain by chain. Bond by bond. The liquid hardens. The cage closes. This is polymerization, the molecular weaving of a stone prison that takes millions of years to complete. But the chemical trap is only half the horror. The real nightmare is what happens to the creature's body inside that cage. The resin does not just surround the organism. It infiltrates it. It seeps into the respiratory system. It coats the exoskeleton. It floods the spaces between cells and replaces the water molecules with polymerizing chains that will, given enough time and pressure, transform soft tissue into a molecular cast of itself. The creature does not decompose. It is chemically arrested. Imagine a forensic X-ray capable of peering through 100 million years of polymer. That is what CT scanning reveals when we look inside amber. The hydraulics of an extinct lung. The articulation of joints that have not moved since the Cretaceous. In one piece, scientists found mites. 0.2 millimeters across. Smaller than a pinhead. The oldest specimens predate dinosaurs by 100 million years. They predate the supercontinent Pangaea. They predate every mammal, every bird, every flowering plant. Amber is not a product of one era. It is a continuous recording device that has run through the worst mass extinctions this planet has ever witnessed. It was running when the Permian-Triassic extinction killed 90% of all species. It was running when the asteroid ended the reign of the dinosaurs. It is running right now. And this is where the open question finds its answer. The dinosaur tail discovered in Myanmar amber, 99 million years old, contained more than feathers. The barbs were there. The barbules. The microscopic structure of plumage that no one expected soft tissue to preserve. But inside the sample, researchers found something that should have been impossible. Iron traces. In exactly the shape and distribution of blood cells. The first assumption was contamination. Modern blood. Laboratory error. But the chemistry did not match. The iron was not from hemoglobin that survived. It was from hemoglobin that was translated. When the tail was trapped, the resin's polymerization process did something unprecedented. It replaced the volatile organic molecules, the ones that decay in months, with stable polymer chains that do not decay at all. The original blood dissolved. But its shape was preserved. Its molecular architecture was copied, molecule by molecule, into a material that could outlast DNA by orders of magnitude. What scientists found was not blood that survived 100 million years. It was a molecular fossil of blood. A ghost imprinted in chemistry. And if that disturbs you, consider what else the resin captured. In one 99-million-year-old piece, scientists found a snail. Not a shell. A snail, with its soft body still visible inside the amber. It had been caught in the exact moment of giving birth. The baby was halfway out. The mother was frozen in the act of creation. The resin did not care about the significance of the moment. It did not select for drama. It dropped at the wrong second, or the right one, depending on how you define preservation. A spider and a wasp locked in combat. A tick with its gut still full of dinosaur blood. A beetle frozen with its bioluminescent organ intact, a prehistoric flashlight that has not glowed in 100 million years. These are not fossils in the traditional sense. They are biological crime scenes sealed in molecular glass. Now imagine the world that produced them. The Cretaceous was not a gentle Eden. It was a green hell. The air was thick enough to taste. Humidity that turned your lungs into saturated sponges. Temperatures 10 degrees hotter than today's tropical forests. A world that sweated. Millipedes longer than a human arm crawled through the undergrowth. Trees bled golden chemical weapons at the slightest wound. Every scratch, every bite mark, every storm-shattered branch triggered the same response: ooze, trap, seal, preserve. The forest was not a habitat. It was a factory of entrapment. And beneath the dripping canopy, there was a sound no fossil can capture. The silence of the trapped. The organisms that stopped making noise because the resin had already reached their lungs. But here is the question we have not asked. We have talked about what was preserved. We have talked about how. But we have not asked the only question that actually matters. Why do we care? Why does a spider fighting a wasp 100 million years ago matter to you watching this, right now, in a world that has nothing to do with the Cretaceous? The answer is this. Amber reveals something we do not want to see. Life is a temporary state of movement. You are defined by your ability to act, to respond, to change. And amber is the only thing in the universe that achieves biological immortality, not through genetics, not through reproduction, but through chemical arrest. The tree did not preserve those creatures to remember them. It sealed them to forget them. To close the wound. To move on. The immortality was accidental. We talk about de-extinction as if it were an engineering problem. We believe we can bring back the mammoth, the dodo, the thylacine. We believe our technology is advanced enough to reverse extinction itself. And yet the most advanced biotechnology lab on Earth cannot do what tree blood does by accident. Perfect, cellular-level preservation of soft tissue. Not a reconstruction. Not a guess. A cast of the original moment, frozen before decay could begin. We cannot replicate it. We cannot improve upon it. We can only study the results and marvel at a process that requires nothing but chemistry and time. Return now to the spider and the wasp. In the beginning, they were a crime scene. A prehistoric showdown. A moment of violence that seemed remarkable because it survived. But consider what that actually means. Those two organisms were not significant. They were not special. They were just two creatures doing what organisms do, fighting for survival in a world that had no idea they existed. And then a tree bled. And the resin dropped. And a moment that should have lasted less than a second became the only thing the universe remembers about either of them for 100 million years. The fight did not make them immortal. The interruption did. So here is the question you take with you. If you were caught in a moment of pure instinct, a fight, a birth, a crime, would you want that moment to be the only thing the universe remembers about you for the next 300 million years? The amber does not answer. It only records. And somewhere, right now, a tree is bleeding.